Okay, good morning, everyone. Today we'll be looking at an interesting topic on hydrocarbons. Today we are looking at factors affecting biodegradation of hydrocarbon. We are looking at biological factors affecting the biodegradation of hydrocarbon. Biodegradation itself, when we say biodegradation, we are talking about the involvement of microorganisms in breaking down hydrocarbons. Naturally, hydrocarbons can break down. They can be broken down by several factors: chemical factors, physical factors, and biological factors. So the degradation of hydrocarbon that involve microorganisms is what we call biodegradation. And we know that microorganisms are involved in breaking down of organic substances and inorganic substances. They can utilize these things for their own growth. So biological factors, they play a crucial role in the biodegradation of hydrocarbons. They influence the rate and extent to which these organic compounds are broken down. And these factors primarily encompass the characteristics of microbial communities that are involved in the biological processes. So we will look at the factors that are involved in this, and we are going to expand these factors and look at the role of microorganisms. Remember, this is petroleum microbiology, and our interest is basically on the aspect of microorganisms as respect to anything that has to do with petroleum, hydrocarbon, crude, the environment, so on and so forth. So the hydrocarbon in the environment, they are primarily degraded by bacteria and fungi, primarily. Although there are so many organisms in the terrestrial and aquatic environment that's involved in these processes, but the proportion of hydrocarbon utilizing bacteria and hydrocarbon utilizing fungi are highly variable. Reason is because not all bacteria are involved in utilization of hydrocarbon. And when we say utilization of hydrocarbon, that means they can use this hydrocarbon for their growth. They can use it as a source of nutrients. So when they use hydrocarbon as a source of nutrient, before they use it, they have to first of all break it down. In breaking it down, they are enhancing removal of the hydrocarbon in form of pollution in the environment for human beings. The hydrocarbon can be a source of pollution for us as human beings. It's a disturbance. But to the organisms, it's a source of nutrient. So it's a win-win for the organism and also we humans, because we need this hydrocarbon to be removed as a form of pollution, and they need this hydrocarbon as a source of nutrient. So, but not all microorganisms are involved in this situation. So we have a variable proportion of this organism in terms of utilization of hydrocarbon. And there's a frequency of reported population of 6% to 82% for fungi and about 0.13% to 50% for soil bacteria, and also 0.03% to 100% for marine bacteria. That is, it's a variable population in terms of organism involved. So, like I said, that bacteria and fungi dominate this. We also have some algae that are involved. We have marine bacteria, soil bacteria, and so on and so forth. Individual organisms can actually metabolize a limited range of hydrocarbons. They can't metabolize the whole type of hydrocarbon. We have different types of hydrocarbons. So not all type of hydrocarbons can be metabolized. Just look at it in the form of human diet consumption, requirements. Some people they are vegetarian. some are non-vegetarians. Some they love carbon-containing food, while some do not. So all these are kind of requirements or appetite or preferences by individual organism. So only a limited range of hydrocarbon substrate can be metabolized by individual organisms. So, but when we have individual organisms with different ranges assembled together in a kind of a mixed population, then we can have overall broad enzymatic capacity that is required to degrade the complex mixture of hydrocarbons, such as a crude oil. You get what I'm saying, right? You understand what I'm saying? Are you with me?
Yes, sir.
Yeah,
An individual organism may not be able to degrade all the types of hydrocarbon substrate. But when we have a population of organisms coming together, so there is a concerted effort, there is a broad enzymatic capacity, each one with its own requirements. They work together as a microbial community. And this microbial community is what gives a faster degradation of crude oil, whether in the soil, whether in the fresh water, whether in marine environments. So the ability to degrade or utilize hydrocarbon substrate is usually as a form of, dependent on the variety of bacteria genera or fungi genera in the environment. The most important hydrocarbon degrading bacteria in marine or soil environment include Achromobacter. I don't know if you have heard the name of before. We have Acetobacter. We also have alkali genes. We have Antrobacter, Bacillus, Flavobacterium, Nocardia, and Pseudomonas species. These are organisms that are usually encountered as hydrocarbon degrading bacteria in both marine and soil environments. Their importance in degrading these sometimes may be limited to their habitats. For instance, Vibrio species is usually found in the marine environment. So that means the ability of Vibrio to degrade hydrocarbon remains largely at the marine environment because that is where its population is. So also, an organism such as Pseudomonas aeruginosa and Bacillus that are found on the soil degrade those that are found polluting the soil environment, and so on and so forth. Sorry, excuse me. In the numerical taxonomy study of petroleum degrading bacteria, it was reported that Pseudomonas, Micrococcus, and Nocardia are members of, and other members of Enterobacteriaceae, Actinomycetes, coniforms, made up of 95% of isolates. I will encourage you to try to know more about this bacteria genera. They are interesting bacteria genera. I know Pseudomonas is popular. Micrococcus, I don't know if it's popular to you, but Nocardia species are not popular to you. You should know these organisms. They are good bacteria that degrade hydrocarbon, and some of them, they have wonderful metabolites that are used in the industry, such as surfactants, antibiotics production, and so on and so forth. Also, we have the Actinomycetes that are even known. They have produced more than 60% of antibiotics that are used clinically. So among the fungi, we have the Eurobasidium, Candida, Rhodotura, Uroblomyces. They are the most common marine fungi that are involved in hydrocarbon degradation. We also have the Trichoderma, we have the Mozzarella, These are the common soil fungi that are involved. Also, we have Aspergillus and Penicillium. They have been frequently isolated from hydrocarbon polluted site. Now, when an organism is isolated from hydrocarbon polluted site, one thing that strikes your mind is this organism is utilizing this for the environment. Let me say, for instance, if we ask you a question, because question can be asked just to, finger your brain, just to see how well you understand the topic. It can ask you like, a fungi was isolated from a diesel polluted environment. Now, we briefly describe the properties of this fungi with respect to petroleum microbiology. Now, what you are expected to say, what you are being asked, is to let you know that this fungi or this bacteria that you're talking about is an hydrocarbon degradation organism. It shows that this organism has a property of hydrocarbon degradation. And further question on degradation, you are able to tackle it on because the organism is isolated from that environment. But if you isolate it from a different environment and you now say this organism can now be used for biodegradation, you are not sure of its ability. But an organism that is isolated from that environment indicates the ability of such a bacteria for that purpose. So in the marine environment, bacteria are generally considered to represent the predominant hydrocarbon degrading element of the microbial community. Bacteria thrive more in the marine environment. So they are considered to be the predominant hydrocarbon degrading element. Fungi are relatively minor component of the marine microflora because fungi require just little water activity than bacteria. So fungi, they prefer environments such as terrestrial environment, like the submerged wood, a surface film of water, and some are probably also prefer some other environments that are not like the marine. But both bacteria and fungi are abundantly present in the soil, and both of them contribute largely to the biodegradation of hydrocarbon in the soil. Now, what are the factors that affect the microbial, biodegradation? I think I have a different slide here, but I'm going to give you this thing on my head. I think I selected a different slide. So what are the factors that affect the hydrocarbon degradation? We have the microbial community. When we say microbial community, we are talking about the composition of the microorganism in the community. They have diverse and abundance of hydrocarbon degrading microorganisms, and they have significant impact on biodegradation. So when we have a diverse microbial community, like I said in the beginning, individual organism with its own preference for degradation can enhance degradation at its own level. Now, when we now have this abundance of individual with different preferences coming together as a microbial community, the degradation becomes successful. Number two is microbial activity. How active are the microorganisms that are involved in this degradation? How active are the organisms? The level of their degradation, are they active or moderately or low? when we say active, we're talking about high rate of metabolic activities, their rate of hydrocarbon degradation processes. How rapid are they? How fast are they? Now, if the microbial community composed of several microorganisms with so much activity, rapid, that means it takes a faster rate for the hydrocarbon to be degraded. But if the microbial community consists of mixed culture of active and inactive, so we are going to have a slow process. That is why, like I said before, when you isolate an organism from an hydrocarbon polluted site, that gives you a clue that this organism has ability to degrade. It has the ability to degrade. And such forms a community, and the community can easily degrade the hydrocarbon. But if we have organisms that are isolated from other sites, and we are talking about non-indigenous microorganisms in this environment, their participation, their roles in degrading this may be slow, because that is not their environment. They are just brought in like a foreign organism, non-indigenous, unlike an indigenous organism. Then number 3 is microbial adaptation. How adapting are these organisms? See, microorganisms can adapt to their environment, including the presence of hydrocarbon, because naturally hydrocarbon can be toxic to other organisms. That is why some of the agents for controlling microorganisms, some of them are from hydrocarbon. Some of them are actually from hydrocarbons, like kerosene. Kerosene, if you apply kerosene on some other microorganism, they will die. Kerosene is a product of hydrocarbon. Now, talk for instance, microorganism adapting to the presence of the toxic substances. So how are they able to adapt to the toxicity of the content of the hydrocarbon determines the rate of biodegradation. If they cannot adapt to it, that is, I'm talking about cell adaptation. That cell has been adapted to withstand or resist the toxicity found in the hydrocarbon in such a way that it does not cause corrosion or cause toxic effect on their cell wall, or on their own growth proliferation. So ability of microorganisms to adapt or prior exposure to hydrocarbon, which enhance their adaptability, can also enhance their ability to degrade this compound. So it will increase biodegradation rate over time. But if microorganisms have never been exposed to hydrocarbon before, or microorganisms are not adapted in the hydrocarbon environment, they may not be able to carry out biodegradation. They may not even survive. We also have the microbial genetics of the organism. The genetic makeup of the organism also play a role in their ability. It plays a role in their ability, the genetic makeup, which is their ability to degrade hydrocarbon. Most species of microorganisms with specific gene that encodes enzymes, the one that can break down hydrocarbons, most organisms in this category are either naturally endowed with this gene or they acquire it from the environment. So if these genes are not present, then such may not occur. Such may not occur. So another factor is microbial interactions. When we say microbial interactions, that is interspecies interactions. Microbes of different genus interacting together to bring about synergistic ability. That is, they come together and work together for degradation. Some can be able to degrade a particular type of hydrocarbon, while the other may be efficient with aromatic, while another may be efficient with aliphatic. Now, coming together, they can both degrade crude oil, because crude oil consists of both aromatic and aliphatic, all types of hydrocarbons. But if they have antagonistic interaction, that is, if their activity can hinder their own proliferation, then it will affect biodegradation. It will affect the breaking down of this hydrocarbon in the environment. So in essence, effective biodegradation of hydrocarbon requires the balance of this organism. Balance of this organism in terms of microbial community. When we have microbial community, active microbial community, the microbial community having ability to degrade, and their activity level is at a rapid stage, and they are able to adapt to the environment, adapt to the presence of hydrocarbon, adapt to the toxic compound, toxic substances found in the hydrocarbon, and also if they have the genetic makeup, that is naturally endowed genes that can help them to break down this hydrocarbon, or ability to acquire genes. There are some organisms that they can actually be non-degrader. For instance, Escherichia coli is not known to be an organism that degrades hydrocarbon. But the gene for degrading hydrocarbon perhaps may be isolated from Pseudomonas aeruginosa, which is an efficient hydrocarbon degrader, and this gene can be inserted into E. coli. When this gene is inserted into E. coli via plasmid, and the E. coli become modified, the E. coli can carry out efficient degradation of the hydrocarbon based on the fact that it has acquired new gene responsible for degradation of the hydrocarbon. And I also talk about microbial interaction among these organisms, which can allow them to, to work together as a kind of synergistic ability, and this allows their degrading ability to function effectively. So, hydrocarbon degradation is a popular activity, a popular characteristics among major bacteria and fungi, as well as other organisms. So algae and protozoa, they are also members of the microbial community, which are found in both aquatic and terrestrial ecosystems, but their extent in the involvement of hydrocarbon biodegradation is largely unknown. Though they are found there, but the extent of their involvement is not well publicized. Although we have some of them that are capable of utilizing crude oil and the mixed hydrocarbon substrates, such as aromatic hydrocarbon and so on, but the availability in the presence of, when we isolate organisms, do not suggest that they have a significant role in degradation. Because when we isolate organisms that are found in degradation of hydrocarbon, the major population that is found there are largely bacteria and fungi. To this end, it makes it that bacteria and fungi, they dominate the presence of organisms that degrade hydrocarbon in the environment. Thank you very much. Concerning the topic of biodegradation, biological factors affecting hydrocarbon.